Hi, my name is Dennis, and this is the eighth in a series of videos on temperament and health. And in this video, we're going to look at phospholipids. In general, the mandala in the center of the temperamental sequences of hot, dry, cold, and moist. A mandala like that can be useful in understanding the formation of organic molecules. Tempering or balancing in the natural order supports the formation of molecules such as alcohol. And alcohol unites the poles of hot, which is in the levity side, and cold, here, hot and cold, which is hydrogen and oxygen in the OH ion. So alcohol represents crossing of two poles. Gums also can be seen as crossing of two poles between dry in the carbon pole and moist. So a gum oscillates between dry and moist. So the substances follow the temporal mandala in the dynamics of their formation and interactivity. But the key to understanding how poles are united is found in the capacity for cell membranes to allow transport between the inner parts of a cell and the environment of the cell. Poor membrane transport results in poor metabolism, and then health in plants and animals and humans is compromised. So I'd like to describe to you the idea of phospholipids by looking at water sol and oil sol. So here we have the root. We're going to move that out. Put water sol here between cold and moist. Water-soluble elements in the plant are very much active in the root and in the forming of a leaf. The most common water-soluble elements are sugars, salts, phenols, flavonoids, and even proteins. So water sol is one side of the temperamental mandala. So I'm going to take the leaf out here. So we go from root to leaf. On the other side, we see hot and dispersing, and that's the realm of the forming of flowers, and then fruits, and eventually seeds. So we could take the oil sol here and put oil sol and water sol as two poles. Between hot and dry, we get the oil components, and between cold and moist, we get the water-soluble components, nitrogen, oxygen, hydrogen, carbon, and we have the temperamental mandala. So if I take water sol here, we could take that cycle going from root to leaf to flower to fruit to seed. That's just the growth of a plant, and it's following very clearly the action of the four temperaments. So now let's take a look at what is a phospholipid. So I'm going to take the water sol here, and I'm going to put it up in the realm of phospho, and I'll take the oil sol and I'll put it in the realm of the lipid. So a phospholipid is a combination of water-soluble elements and oil-soluble elements. Down below, we see here, if I took this little form here, and I put water sol down below and oil sol up above there, the little tail up there is an oil-soluble element, and the little circle is a water-soluble element. We could say that's the smallest particle of oil suspended in the smallest particle, or near the smallest particle of water. Oil sol and water sol happen very often in the forming of whatever it is, hydrosols, through distilling essential oils. So in a hydrosol, you have water sol and oil sol, but actually these little units of water and oil soluble are everywhere. They're all in the blood. But what they have a tendency to do is the water-soluble elements have a tendency to link up, and the oil-soluble elements have a tendency to link up. And when they do that, they form a membrane. They form a phospholipid membrane. But, the oils are going to be attracted to each other, surrounded by water droplets. So actually, the most fundamental form is something called a micelle. And we can see in the forming of the micelle that the outer rim of the micelle is the phosphate water-soluble side, and the inner oil side that are attracted to each other is the lipid side. So the micelle is the most fundamental unit of the phospholipid. But when many micelles get together, they start to link up in layers that are known as bilayers. So down here, down below at the bottom, we see the universal bilayer cell membrane. That's known as a phospholipid bilayer membrane. So here's why. So the water-soluble phosphate parts are on the outside, and they form a layer, and the lipid-soluble parts are on the inside. They form kind of like an Oreo cookie with the fat in the water. The fat molecules link up, and the water molecules link up, and they form a bilayer with a little layer of fat in between. Now that form there makes use of salts like calcium and sodium and potassium in order to make rigid structures that we call cell membranes or cell walls. And if all there was the phospho and the lipid, then the cell walls would be impermeable because we'd have all the oil linked up with each other, and they would all kind of connect up and just kind of grind up into each other. But we don't have that. We have a situation where the lipids are separate from the phosphorus elements, and so they form a kind of very unique boundary. And that's really good because every cell and every organelle in every cell separates itself from the environment, from the outside and the inside of the cell through a phospholipid bilayer membrane. So then how do things get inside and outside? So let's put our phospholipids up here. Let's put phosphorus down here, and we'll put potassium over there. We'll put our lipids up here, and we'll put a protein up there. So what we've done there is divided the temperamental mandala into the water-soluble salts of potassium and calcium and phosphorus and sodium. They are known as potassium or calcium pumps because they pump those salts in and out of the cells to regulate the way in which the cells interact with the fluids inside and outside of the cell. But in order to do that, they need a gate to open to allow the potassium pump to work. The gates in the phospholipid bilayers are made from proteins. And the proteins are up here in the moist and flow category. There are diagrams of these online that are actually kind of funny. They actually look like a gate that's kind of rotating like a turnstile in the subway. Chemically, the potassium and sodium ions are flowing through the way in which the proteins close the gate and open the gate and close the gate and open the gate, allowing the salts to go in and out through different walls in the cell so we can have a permanent semi-permeable membrane that's continually flowing between the outside of the blood and fluids outside the cell and what's inside the cell, because the protein gates are continually opening and closing based on the way in which the salts are regulating phosphorus and potassium. Now this is important because down the road, that phosphorus and the protein need to come together in a compound that is extremely important for life, and that is as adenosine. Adenosine is a protein structure, we'll put it up here with the proteins, that has a very special relationship to phosphorus because it creates a substance known as adenosine triphosphate, known to the world as ATP. ATP is the energy component within cells, having to do with how life is continued, how cells metabolize, how they live, how they grow, how they die, and how fluids move through the body. So phosphomembranes and the phosphorus in them connect up to the protein structures in body fluids, and ATP is the key that unlocks the protein gates that allow the potassium pumps to refresh the inside of a cell and the outside of a cell and regulate fluids in the body. Thanks for watching.